Good evening, ladies and gentlemen. In today's video presentation, we will be discussing gene expression and regulation. To kick off our presentation, I would love to introduce our wonderful presenters. Alyssa DeLeon, a psychology major, Desiree Duncan, a biomedical science major, Jaedyn Johnson, who is currently double majoring in biomedical science and chemistry, and Jorgenson, who is currently majoring in coastal environmental science. So without further ado, let us begin. Today, we will be introducing gene expression and regulation and how it relates to Mendelian and molecular genetics. We will be discussing what each topic is and how they work, then relating each back to one another, in addition to some questions to make sure you are all grasping the concept. Today, we will be introducing gene expression and regulation and how it relates to Mendelian and molecular genetics. We will be discussing what each topic is and how they work, then relating each back to one another, in addition to some questions to make sure you're all grasping the concept. On today's agenda, our presentation will be described by discussing 4 key points, a summary of the information, and some questions to make sure you're all listening. The things going on within our body can be compared to an orchestral symphony. The way the orchestra comes together in harmony in order to construct a proper tone and sound is relative to how the organisms and elements within our body interact with one another in order to function and send signals. In our first slide, we will be discussing gene expression and regulation. Gene expression is the process by which the information encoded in a gene is used to direct the assembly of a protein molecule. The process of transcription and translation are seen through gene expression and regulation. Transcription is a process of which information in a strand of DNA stores genetic material inside the nuclei of cells into a new molecule. Translation is a process of creating proteins from an mRNA template. In order to control the rate at which a protein of RNA product is produced, genes must undergo gene regulation to determine when and where specific genes are initiated. As proteins and RNA products are produced, many genes are expressed during either the process of cell differentiation or as a result of cell differentiation. Gene regulation refers to the mechanisms that act to induce or repress the expression of a gene. We will discuss transcription and translation at a deeper level later in the presentation. To further understand Mendelian genetics, to continue, we will now introduce the topic that is Mendelian genetics. So what is Mendelian genetics? Mendelian genetics comes from an Austrian monk named Gregor Mendel, who introduced a new theory of inheritance based on his experimental work with pea plants. He conducted these experiments to prove that what we inherited from our parents was not a blending of the two, but rather that heredity is the result of discrete units of inheritance, and through this, every gene was independent in its actions in an individual's genome. To understand genetics, he conveyed that when looking at different genes, they each come in pairs and that they are all inherited as units from each parent that are carefully tracked through genotypes and phenotypes. The 3 basic laws of Mendelian genetics that he introduced are as followed: the law of independent assortment, the law of dominance, and the law of segregation. Through a careful study of genetics, he understood that the law of independent assortment states that different pairs of alleles are passed onto the offspring independently of each other. Therefore, inheritance of genes at one location in a genome does not influence the inheritance of genes at another location. The law of dominance states that since there are dominant and recessive alleles, only one will remain dominant and will be expressed with the phenotype. The law of segregation states that for any trait, each parent's pairing of genes or alleles split, and one of these genes passes from each parent to an offspring. Which particular gene in a pair gets passed on is completely up to chance. To further understand Mendelian genetics, gene expression and regulation can be seen when transcription and translation are introduced. Within Mendelian genetics, transcription is seen as the process of making an RNA copy of a gene sequence. This copy, called a messenger RNA, or mRNA molecule, leaves the cell nucleus and enters the cytoplasm, where it directs the synthesis of protein, which it encodes. Translation is the process of translating the sequence of a messenger RNA molecule to a sequence of amino acids during protein synthesis. The genetic code describes the relationship between the sequence of base pairs in a gene and the corresponding amino acid sequence that it encodes. To further this presentation, we will be discussing molecular genetics. What is molecular genetics, you may ask? Molecular genetics, introduced by Niels Bohr, is the study of the molecular structure of DNA, its cellular activities, and the influence it has on determining the overall makeup of an organism. With molecular genetics, we were able to truly prove what Mendel was observing when studying his pea plants due to the modern technology that has been developed since Mendelian genetics was first introduced. The biggest factor that has helped improve these findings and give us a true answer rather than observations is the invention of the microscope. Throughout molecular genetics, somatic cells, a non-reproductive cell of an organism, contain strands of DNA that are carefully arranged in chromosomes. Therefore, in order for the DNA strands to be carefully packed into the chromosome, they must undergo 2 levels of coiling. The first level is coiled around the central core of the 8 histone proteins, resulting in nucleosomes, those in which are connected by spacer DNA. The second division of coiling is constructed when the DNA double helix begins to coil around a string of nucleosomes and a chromatin fiber. Similar to gene regulation in Mendelian genetics, molecular genetics also undergoes its own variation of transcription and translation. As we already understand the definitions of translation and transcription, we will now identify how they too apply to molecular genetics. Within molecular genetics, a transcription machinery is bound to its gene promoter, and in order to synthesize a single strand of mRNA molecule base by base, so it is complementary to the sequence, the double helix must open. To follow this, the promoter and enhancer elements are involved in gene transcription to an additional mRNA molecule in order to give mature mRNA. Furthermore, translation then allows mature mRNA molecules to be translated to proteins. As the mRNA splits, the mRNA can be read as triplet codons, which allows for each codon to correspond to a specific amino acid. Codons can finally be translated after the amino acids link into a growing peptide chain in order to fold into their formation. To relate all the ideas to each other, let us look at how similar they are. In both Mendelian and molecular genetics, we can see that the big difference between the modern science of genetics and Mendel's basic laws is that modern scientists have a much clearer understanding of the mechanisms behind the patterns Mendel observed. Additionally, most of Mendelian genetics was solely based on observations of offspring through generations in hopes that Mendel was able to observe these changes, aka his pea pants, easily. Now we are able to look into the genetic makeup of these plants with the help of molecular genetics. In a similar fashion, we can see how within every principle, transcription and translation prove to have a grand effect on the overall outcome of the situation. Whether it be creating proteins or the assembling of a peptide chain, they all correlate back to the same need. All right now, don't get too nervous. Let's see what you can remember and if you paid any attention to the previous slides. Question 1. What are not one of the 3 laws introduced by Mendelian genetics? The law of independent assortment, the law of regeneration, the law of dominance, or the law of segregation? And the answer is B, the law of regeneration. If you answered B, you would be correct. The 3 laws of Mendelian genetics are the law of independent assortment, the law of dominance, and the law of segregation. Alrighty now, next question. Question 2. What is the similarity between gene regulation and expression in Mendelian genetics and molecular genetics? Okay, if you said gene regulation and expression is shown in Mendelian genetics by making proteins which allow for a difference in alleles, while in molecular genetics, genes are expressed and regulated by translating mature mRNA molecules into proteins, you would be correct. Furthermore, to conclude our presentation, without these improvements in modern technology, all that we would know would be mainly based on observations and common theories. Within life, each cycle is repetitively activated within the cell, which thus produces a healthy established body, and the music that is life continues to develop. Thank you for enjoying our presentation, and we hope you have a wonderful evening.